Hello everyone, welcome to Biotechnika's YouTube channel. So today we all will be learning about abscisic acid phytohormone, where you have to learn some important points like discovery, biosynthetic mechanism, signaling pathway, regulation, transport, as well as functional profile of plant growth regulator, that is ABA. So let's come on to the topic and understand all these points one by one. So firstly, coming to the introduction of this phytohormone. So it is a ubiquitous hormone that is found in vascular plants. So what are vascular plants? The plants that contain xylem and phloem in it. So it is a 15-carbon terpenoid compound that belongs to the class of secondary metabolites, that is terpenes. So when terpenes are oxygenated, they forms terpenoid. So this abscisic acid, it is 15 carbons in structure. That's why it is also called as sesquiterpenoid. It is also regarded as a stress hormone as it regulates various types of abiotic stresses in the plant cells. So it is also regarded as a stress hormone. It is synthesized in the vascular tissues of the plant, that is xylem as well as phloem, where it regulates salinity, dehydration, as well as temperature stress, where it regulates stomatal opening and closing, means it is regulating various types of abiotic stresses in plants. So for the first time, it was identified in 1916 as a growth inhibiting substance, means as a substance that is inhibiting the growth of the plant. Now what's the growth promoting substance? It's brasinosteroid. And what's the growth inhibiting substance? It's ABA. Because it was seen that it was associated with the onset of bud dormancy, means it was keeping the bud dormant, means there was no germination and the bud was not getting germinated, means it was in a dormant state. No germination means growth inhibition. And also it was promoting the abscission of cotton fruit. What's abscission? It's a natural detachment of any fruit or leaves from the plants. So if the plant or fruit will be detached, will it be germinated? No. Means the growth is inhibited there. That is why it is called as the growth inhibiting substances, where it was seen with the onset of bud dormancy as well as the abscission of the cotton fruit. For the first time, it was discovered by Friedrich M. Endicott. So this scientist is known for the discovery of abscisic acid. Now let's see the biosynthetic mechanism of this abscisic acid. So this abscisic acid, it is synthesized in almost all the cells that contains chloroplast or amyloplast. So both these chloroplast and amyloplast are plastid. That contains chlorophyll is called as chloroplast and the stores starch is called as amyloplast. Now what's the precursor for the synthesis of this ABA? It's a 40-carbon carotenoid intermediate, that is what called as zeaxanthin. So this zeaxanthin is only responsible for the production or the synthesis of this ABA hormone, that is abscisic acid. What's the location? The early step of this synthesis, it is happening in plastid, as you can see here, and the final step is taking place in cytosol. So two organelles are involved, one is plastid and one is the cytosol. And what's the name of the pathway which causes the production of this abscisic acid? Is isoprenoid pathway, that is IPP. So the pathway starts with the zeaxanthin. It's IPP pathway which starts with zeaxanthin, that's the precursor for this ABA. It's a 40-carbon precursor that is converted to voilaxanthin and then to neoxanthin. So all the three intermediates, they are formed in the plastid. As you can see here, they are 40-carbon intermediate. So you can just remember a code like ZVN, that is zeaxanthin, voilaxanthin as well as neoxanthin. So they all are 40-carbon precursor. Now once the neoxanthin is formed, it is diffused out from plastid into the cytosol, where it is converted to xanthosine. So you can see here, this is converted to xanthosine, which is a 15-carbon intermediate. And we have seen that abscisic acid is a 15-carbon compound. Means this xanthosine will help in the production or the synthesis of abscisic acid. So this xanthosine, which is a 15-carbon, it is now converted to ABA aldehyde, again which is a 15-carbon compound, which is then converted ultimately to ABA. So the aldehydic group is removed and it is converted into ABA, which is again a 15-carbon. So for this you can just remember the code, that is XA square, that is xanthosine, abscisic acid, aldehyde as well as abscisic acid. These all are 15 carbon. So the final step, it is catalyzed in cytosol. Now what's the regulation of this pathway? So firstly, this ABA is inactivated if its concentration exceeds the optimal level. So inactivation is done by oxidation reaction where the enzyme involved is ABA 8 hydroxylases. So it will oxidize this abscisic acid and this is now inactivated. And the next is ABA inactivation by conjugation, where this ABA is conjugated by some other means and it is inactivated inside the plant cell so as to maintain the optimal concentration of this abscisic acid phytohormone in the plant cell. Now let's see the transport mechanism. As we have seen that this abscisic acid, it is ubiquitous to all kind of plants that has vascular bundles like xylem and phloem. Means this is synthesized in the vascular bundle like xylem and phloem. So it will be transported from xylem as well as phloem both. Now we all know that xylem is involved in the transportation of both water as well as solutes. So with the help of xylem, this abscisic acid is transported. Now what if this abscisic acid is synthesized in phloem? So it will be transported via help of phloem sink stream. So if it is in phloem, it will take the pathway of phloem sink, and if it is in xylem, it will be transported along with water as well as solutes. Now let's see the signaling mechanism of this abscisic acid. So once this abscisic acid is synthesized in the plant cell, that is xylem and phloem, it initiates the signaling cascade. And here the signaling includes phosphatases as well as kinases. What's the name of phosphatases here? It's PP2CA. And what's the name of kinases? It's SNRK2. What's the function of phosphatases? That will remove phosphate group from the target. And what does kinases do? It will always add phosphate group to its target. That means they both are antagonists to each other. What's the ligand here? It's ABA itself, that is a phytohormone that is synthesized via the biosynthetic mechanism. What's the name of the receptor? Pyrabactin resistant, or you can say it's PyL, that is pyrabactin resistant like one, and the last is RCAR, which is regulatory component of abscisic acid receptor. So all these three receptors can be acting as a receptor for the ligand, that is abscisic acid. So what happens? All these three receptors belongs to the family of start domain. So they have a start domain in their structure, and to this start domain only the ligand abscisic acid will bind. So what happens if the abscisic acid is absent? If it is absent, it will not bind to the receptor. Here only one name of the receptor is mentioned, that is PYL. You can consider it like PYR, PYL or RCAR. It can be any of three. So if PYL is free, that means abscisic acid is not present. If this is not present, so this phosphatases will be activated here. What's the name of phosphatase? It is PP2CA. Once this is activated, it will remove the phosphate group from its target. And what's the target for this phosphatase? It's a kinase, that is SNRK2, which is called as sugar non-fermentin related kinase 2. It will remove the phosphate group from this kinase enzyme. And if the phosphate group is removed, suppose from here phosphate group is removed, so what it will do? This will not be able to phosphorylate its target protein. Now what's the target? It's a BZIP transcription factor that is involved in the abiotic stresses like drought stress and also involved in the opening and closing of stomata. Means no abscisic acid induced expression will be seen inside the plant because abscisic acid is absent. And here you can see phosphate group is added to this kinase. Then how come it is added to this kinase? Because SNRK2, it has a autophosphorylation ability where it can add phosphate group to itself. But if the phosphatase is activated, then it will remove this phosphate from the target, that is SNRK2 kinase. Now what happened if this abscisic acid is present? If this is present, it will always bind to its specific receptor that can be PYR, that is PYR or RCAR. Now the receptor is activated. Once the ligand binds to the receptor, it will induce a conformational changes in the receptor that will open up a binding pocket for the phosphatase. Now what's the phosphatase? It is PP2CA. Now this has bind to the receptor. So once this phosphatase is binding to the receptor, this is inhibited. Now the phosphatase activity is suppressed. Now it will not be able to dephosphorylate the target, that is kinases. So now here kinases is free from any kind of phosphatases, or it will be able to autophosphorylate itself. Now, if this phosphorylation is happening, it is able to phosphorylate its target also. Now, what's the target? That is B-zip transcription factor that is involved in the abiotic stresses like drought as well as the stomatal opening and closing. That means phosphate group will be added to the target, to the transcription factor, and there will be a ABA-induced gene expression in the plant. So how this ligand and receptor are interacting with each other? It is because of the start domain which is present in the receptor structure. So this is how the signaling is happening. So if the abscisic acid is absent, phosphatase will be activated. That will remove the phosphate group from the kinase, that is SNRK2. And this kinase will not be further able to target or to phosphorylate its receptor protein or target, that is B-zip transcription factor. But what if ABA, that is abscisic acid, is present? It will always bind to its receptor, that is PYR or RCAR, which will induce a conformational changes in the structure and will cause the interaction of phosphatases in an ABA dependent manner. And now phosphatases are inhibited and this kinases are free. That will causes the phosphorylation of its target protein like B-zip transcription factor and will induce the ABA-induced gene expression in the plant. That will cause the abiotic stresses like drought as well as involve in stomatal opening and closing. Now let's see the function of abscisic acid. So this abscisic acid is very well known for the function, that is vivipary. Now what is vivipary? Vivipary is a condition in which a embryo is attached to the maternal plant and results in the continuous growth. Means you can see the continuous growth of the embryo plant still when it is attached to the maternal plant. This is what called as vivipary. So it helps in the germination of seeds in the fruit when the embryo is attached to the plant. Means it is still attached to the plant, that is vivipary. That's why it is called as sprouting of the seed, but still in the pre-harvest condition. That's why it is also called as pre-harvest sprouting. It is a characteristic of grain crops, like you can see this vivipary in wheat, barley, rice as well as sorghum. But for the first time, this vivipary was studied in maize, which was shown that it requires gibberellic acid as a positive signal. So if abscisic acid is present in the plant cell, it can induce vivipary, which is the main function of abscisic acid. But for this function, it always requires gibberellic acid as a positive signal. Means gibberellic acid or gibberelline phytohormone has to be there so as to induce the vivipary with the help of abscisic acid. So this is the main function of abscisic acid in the plant. So what we have seen? We have seen about the abscisic acid, where we have seen it is a hormone that is found in the vascular bundle like xylem and phloem. Then we have seen its discovery, that was discovered by Friedrich M. Adicott. We have seen the biosynthetic mechanism where it is taking place firstly in the plastid and then is the cytosol. What's the precursor? It's a 40 carbon carotenoid precursor, that is zeaxanthin. And also we have seen the function profile, that is vivipary. We have seen the signaling pathway, which is the phosphorylation and dephosphorylation pathway, which involves the use of phosphatases like PP2CA as well as kinases like SNRK2. As well as we have seen the transport with the help of xylem as well as phloem sink stream. So this was all about the hormone, that is abscisic acid. So I hope this topic was clear to everyone. If you like the video, do not forget to like, share and subscribe to the channel, that is Biotecnika. Meet you in the next video. Till then, take care. Bye-bye. Happy learning.